Dear Falling Walls Lab Slovakia finalists, dear members of the jury, dear distinguished guests, let me welcome you all cordially at the Falling Walls Lab Slovakia 2024 final. My name is Hanna Overweten-Rapantova and I will guide you through today's event. And now let me introduce to you the organizers. President of the Falling Walls Lab Slovakia, Professor Peter Moco, academician of the Learn Society of Slovak Comedians in Bratislava, jury chairman, Falling Walls Lab Slovakia, Professor Vladimír Búžek, academician of the Learn Society of Slovakia, Slovak Academy of Sciences, project manager, associate professor Marian Holienka, Comenius University in Bratislava, and the project team, Mrs. Diana Suchankova and Mrs. Eva Ručeková, Comenius University in Bratislava. I would like to also welcome our distinguished guests, Dr. Robert Ševčík, General Director of the Research and Development Section at the Ministry of Education, Research, Development and Youth of Slovak Republic. And I would like to also welcome our guests from academia, Associate Professor Eva Viglašová, Vice Rector of the Comenius University for Education and Social Affairs, Professor Ljubica Lacinová, member of the Presidium of the Slovak Academy of Sciences, Engineer Lubomír Bilský, Executive Director at Slovak Organization for Research and Development Activities and main organizer of European Researchers' Night. And it is my pleasure to welcome Dr. Zuzana Veziak from DAAD, the German Academic Exchange Service, the lecturer at Economical University of Bratislava. Welcome. And now, ladies and gentlemen, allow me to invite seismologist Professor Peter Moco from the Comenius University of Bratislava, the president of Falling Walls Lab Slovakia 2024, for his welcome and introductory address.
Ladies and gentlemen, welcome to 7th national event of Falling Walls Lab, as we call it, Falling Walls Lab Slovakia. So I have to do something against this light. I will go, no, another light. It is terrible. So I will be not looking at you, but downstairs. Let me first explain the story of Falling Walls Lab Slovakia. The infamous Berlin Wall separated Western Berlin from Eastern Berlin since 1961 until 9th of November 1989. 9th of November is considered the day when the Berlin Wall fell. Strictly speaking, on that day, demolition of the infamous wall started. The infamous wall was really one of the best known symbols of political division between West and East. 20 years later, one of the leading quantum physicists in Germany, Professor Jürgen Mlinek, got the idea to create a unique worldwide opportunity for young researchers and innovators and organize Falling Walls Lab. Falling Walls of some unsolved problem. And the first event happened on 20th anniversary of the world-changing event of Berlin Wall. Unfortunately, at that time, we were somehow not aware of this, and for 8 years, Slovakia was not present at the final in Berlin. So about 100 participants, young researchers and innovators, were gathering together every year, and there was absolutely no mention about Slovakia. This was changed in 2017, thanks to Professor Vladimír Bužek, because at that time Professor Vladimír Bužek was one of the leading organizers of one big quantum flagship project, and Professor Jürgen Mlýnek, who established Falling Walls Lab in Berlin as a world event, came to Bratislava to meet Professor Bužek and to meet at that time Minister of Education. I was happy and lucky when Vlado Bužek told me that we could have dinner together with Professor Jürgen Mlýnek, and we developed the idea of Falling Walls Lab Slovakia, simply in order to have Slovakia present at the Berlin final every year. And this was true every year since 2017 until except 2019. For some technical reasons, we didn't organize this. So the winner of Falling Walls Lab Slovakia represents Slovakia at Berlin on 8th of November. It is a prestigious event, and the jury in Berlin final always has at least one or two Nobel Prize winners and very distinguished, very important and significant scientists and innovators. As you know, each participant has exactly 3 minutes to present his idea, project, vision, or result, or recipe, or solution. 3 minutes are relatively short time, but it is relative. 3 minutes are very long time, sufficient time for earthquake to destroy cities and to kill even hundreds of thousands of lives. 3 minutes are very critical in the case of heart or brain attack. At the same time, 3 minutes, as I said several times on this occasion, were enough for creation of everything substantial in our universe. You are probably in better position for 3 minutes presentation because you will summarize results of your long-term or simply some time-lasting research and effort, and you are probably better ready than something which created our universe from nothing. So I hope that all we can enjoy this event. We will be listening and watching your presentations. We are very curious what this year will bring, and we hope to select in the jury the best potential representative of Slovakia for this year Berlin final. So enjoy this event. Thank you for coming. Thank you for the attention.
Thank you very much, Professor Peter Moco, for your welcome address and encouraging words. So we are all very excited about today's afternoon full of bright innovators. It is my pleasure to announce the next welcome address. The Falling Walls Lab Slovakia has been always supported by the German Federal Foreign Office and DAAD, the German Academic Exchange Service. Therefore, I am very happy that Dr. Zuzana Vesiak is going to share some words with us.
It's good I have a paper in my hands. Yeah, after this historical overview, I am even more happy to be here, actually already the 5th time in a row. So yeah, to be part of such a awesome event in Slovakia. And I will just say a few words about the Falling Walls Lab. It is a global platform organized by the Falling Walls Foundation. And as you also said, founded by the German Federal Ministry of Education and Research. It is supported by the Federal Foreign Office of Germany, the German Academic Exchange Service, and the Ministry of Education, Science, Research and Sport of the Slovak Republic. And it brings together a diverse and interdisciplinary pool of students, researchers, early career professionals by providing a bridge, which we heard, it's kind of the historical meaning of it, to break through ideas globally and also locally. And these events each year from March to October bring together or showcase the quality, diversity and passion of the region's most innovative minds. So I am very excited to hear the finalists this year, and of course at the end to announce the winner and to give the presents. Thank you very much, and I hope you will enjoy this event.
Thank you very much, Dr. Veziak, for your supporting words. Since 2017, when Falling Walls Lab competition was introduced here in Slovakia, quantum physicist Professor Vladimír Búžek from the Slovak Academy of Sciences has been the jury chairman of Falling Walls Lab Slovakia. So I would love to invite now Professor Búžek to the stage to introduce the jury members, the rules, and the evaluation criteria for today's competition. Welcome.
Okay, so before we start, I'd like to introduce you the jury. The jury is composed of distinguished scientists in Slovakia, very distinguished, but I take a liberty not to read all the academic titles, it would be too long. So I just would like to welcome Professor, okay, I said no titles, I'm saying titles, Silvia Pastorekova, Katarina Mikušová, and now in alphabetical order, Petr Celec, Marian Holienka, Robert Mistrík, Peter Moco, whom you already know, Ivo Petráš from Košice, and Jan Svák. Thank you. So I'm really very thankful to the jury members that they found it possible to come and to judge your presentations. Now, about your presentations, you know, this is very unusual type of event because these are, okay, so these are elevator speech. Okay, this is horrible, but okay. These are essentially elevator pitch speeches, very short, 3 minutes. I mean, the whole point is that in 3 minutes, if you have something to say, if you're enthusiastic about what you do, it's enough for you to have 3 minutes to present it and to attract other people to what you are doing. So that's the point, 3 minutes. And after that, because I'm certain that your presentations will be interesting, you have 30 seconds for potential questions that will be asked. Okay, so this is the point. 3 minutes plus 30 seconds, and you go off. That's very strict. I mean, we are not going to make any concessions here. So that's how it will look like. The second thing is that the jury will somehow score or evaluate 3 criteria. We will use 3 criteria. You will have the 50% of your, whatever the number of scores is, but 50% is for the breakthrough factor. How original, how innovative, how exciting your idea you are presenting is. The 40%, next 40%, is for the relevance and impact. I mean, how the society will be impacted by your idea. It's very important that what we scientists do have a social impact in one or other way. And therefore, it's such a, so another criterion for 40% of that is for relevance and impact. And finally, the 10% is for the performance. Okay, we are not Hollywood actors, so I think that therefore it's just 10%. But it's important also for scientists to be able to present what you have to say. But so we will weight whatever your performance is by 10%. So don't be nervous. Focus on the first 50%. That's the most important. And finally, there are 2 points finally. The first point is that you probably noticed that we teamed teamed with the European Researchers' Night. Via this, we hope that we will create bigger impact of this falling wall slabs competition. And the Researchers' Night is tomorrow, so the day before it's today. We have this meeting, we have this conference, if you wish. And the final thing is really the final one. I'm impressed. I'm really impressed. Out of 17 finalists, there are only 3 men. I mean, this is impressive. I never ever seen, I mean, I'm from natural sciences, so maybe in social sciences the situation is different, but in natural sciences I have never seen this ratio between speakers, men and female speakers. So I'm very thankful to all of you who came here to make this big difference and that young women understand how science is important. So thank you. I wish you all good luck and probably we'll start soon.
We will start soon. Thank you very much, Professor Bužek, for explaining the criteria and also for introducing us, the jury members. And now we really can proceed to the presentations themselves. As you could hear, we have 17 competitors, which will follow in alphabetical order. Our finalists, I'm going to repeat it one more time, are going to present their innovative results, projects, or ideas in exactly 3 minutes. Pitches, as Professor Bužek has mentioned, and I will stop any discussion after the time has run out. First, it is my pleasure to announce a presentation of Zuzana Bartová from the Slovak Academy of Sciences, who is going to present her idea that breaks the wall of environmental pollution. Here we go.
Most of the pollution we face now is a legacy of older industrial activities. Tailing ponds, heaps, mine-impacted waters. All these sites are full of unused metals posing a significant threat to the environment. On the other hand, organic pollutants are mostly present in chemicals that spread around, whether they remain on the surface or are stored in unsuitable containers. But besides that, all those problems represent also a huge opportunity. Well, let's explore some possible solutions here. Many contaminated sites are rich in valuable metals, some of which are classified as critical raw materials essential for supporting an independent circular economy within the EU. Achieving our goals requires modern approaches tailored to each polluted site individually. Solutions we offer are in so-called biohydrometallurgy and electrochemistry, depending on the nature of the pollution we face. Biohydrometallurgical processes allow us to extract metals from liquids using environmentally friendly methods such as bacteria. After bioleaching, metals can be found in what's called pregnant leachate, and from this we can recover them, for example, like this. In case of organic pollution, the best results have been achieved using electrochemistry, where organic pollutants are broken down using a special boron-doped diamond electrode. This process is really unique because it combines the BDD electrode with several downstream steps, and the whole process has already been patented. This green, low-cost technology allows us to break down organic pollution into carbon dioxide, water, and inorganic products that can be treated separately. Pollution can be degraded in a matter of hours, often reaching potable water quality. Moreover, this process is so efficient that it can even be powered by photovoltaics. Well, while the waste and byproducts of the past may not directly be our fault, we should view this as an exciting chance to develop new technologies. Thank you.
Thank you very much. Thank you very much, Zuzana Bartova. If somebody has a question, now it's the time. We have a couple of seconds left. Is there a question from the jury? Or from
This is a very general global problem.
Yes.
How unique is your approach compared to other approaches? Because I know that there are many approaches to recycle, if you wish, this pollutant.
Yeah, we are trying to prepare this technology that is customized to individual environmental pollutions. So, for example, we are focusing on Nižná Slaná, and we come there and we are trying to build a pilot there. At first, we do the research at lab, and we work with the natural waters right from there. So many researchers in general work mostly with synthetic solutions, and we are trying to work with the natural samples. So it's like much more efficient, let's say.
The other point is that what you just said, that you are tailoring the recycling.
Exactly.
For the specific problem.
To the specific pollution, yes, yes.
Thank you.
Thank you very much,
Thank you.
Zuzana Bartova. Our next presenter is Emil Bečka from Comenius University Bratislava, who will present his idea that breaks the wall of trauma patient infection.
Thank you. Have you ever wondered what happens with your immune cells during the trauma injury? So, after vehicle accidents, surgeries, falls, or basically, or in general, after traumatic injuries, your damaged tissue and cells release their intracellular particles, including mitochondria, which can activate and attract immune cells. Activation of immune cells is based on mitochondrial similarities with bacteria sharing similar structure and molecules, and these similarities comes from their shared evolutionary ancestry or origin, when the mitochondria and bacteria were closely related. But during the time, mitochondria became a part of almost all eukaryotic cells, serving as a power plant and supporting almost all cells with energy and also hiding themselves from immune reaction. But the problem is that once our mitochondria are released during the trauma injury, they can activate the new immune cells, which, when they are already activated, they cannot respond properly to possible bacterial infection, which is very common in patients after traumatic injury, which are hospitalized in hospitals. So let's imagine that immune cells can recognize just one trigger, in our case, mitochondria, but they cannot respond or react or recognize another more dangerous, such as more dangerous things, such as bacteria. And another problem is that most of the drugs using or administered after traumatic injuries aimed on total immunosuppression, leading to blockation of immune cell response towards both mitochondria and bacteria. Our approach takes a different way, suggesting that using activator instead of blockator of immune cells can lead to modulation of their response towards mitochondria, basically ignoring their presence from the cell and focus their reactivity towards the bacteria. So, to conclude this, administration of these activators can prevent reaction of immune cells or immune response towards the mitochondria after the injury and focus their response just towards the bacteria in the patients after hospitalization. Thank you.
Thank you very much. Thank you very much, Emil Bečka, for your presentation. Any questions, please? Now it's short time. Professor Pastorekova.
Are there any similar approaches used already in clinic, or it's totally innovative?
It's a tricky question. I don't know so much about these, but most or the first aid is using immunosuppressants as cortisols and these things. And I don't know much about these activators of immune response.
So probably it's not there, and it's a novel approach.
Yeah, that's that innovative thing.
Thank you.
Thank you very much, Emil Bečka. Now it is my pleasure to announce a presentation of Katarina Bilska from Comenius University Bratislava, who is going to present her idea that breaks the wall of biofilm-associated infections.
Thank you. More than 80% of chronic infections are connected to biofilm-associated infections, which are a major challenge for clinicians. Microorganisms form biofilms on the surface of implantable and non-implantable medical devices, such as catheters and endotracheal tubes, and they are responsible for many diseases, for example, urinary infections, endocarditis, or other types of infections. The most of these biofilms are resistant to conventional therapeutics. It is therefore crucial to find alternative methods how to combat with biofilm-associated infections. Our goal is to produce photoactive nanomaterial with anti-biofilm properties suitable for medical devices. The basis of material is polymer, for example, polyurethane, commonly used for medical devices, modified by a layer of synthetic claymyl saponite, functionalized with a photoactive compound, floxin B. The surface of this material has antimicrobial properties, and still we can add other antimicrobial compounds to have more effective material. After irradiation with a green laser or a green LED lamp, reactive oxygen species are formed which damage DNA, proteins, and membranes of bacterial biofilm cells. We tested this material on gram-positive bacteria, Staphylococcus aureus, on sensitive and resistant strain to commonly used antibiotics. And we achieved more than 70% of inhibition on this material compared to non-modified polymer. After irradiation with a green laser for only 2 minutes, we achieved more than 99% of inhibition of biofilm cells. Important part of research is about mechanisms of bacterial cells, how they react on this material. For example, if efflux pumps or adhesins affect effectiveness of this material. These understandings can help improve this material, and our results showed that this new design, nanomaterial, is a useful tool to development of medical devices such as catheters, endotracheal tubes, or wound dressings. Thank you for your attention.
Thank you very much, Katarina Bilska. Any questions? Professor Celet?
My question would be, okay, these materials act against bacteria, but what about the effects on the host human tissues or cells?
We tested compartment of this material because of toxicity, for example, and there are no toxicity. We prepare in vivo experiment on mice if there are some problems. But, for example, floxin B as an important part of material is approved by FDA for food coloring, so this compound is really safe, and saponite too, so this shouldn't be a problem.
Thank you very much, Katarina Bilska.
Thank you.
Our next presenter is Martina Bugriová from Slovak Academy of Sciences, who will present her idea that breaks the wall of an iodine isotopes.
Thank you very much. I'm from Comenius University in Bratislava. So why iodine? Iodine is a radionuclide which occurs naturally in the environment and also can appear as a contaminant. The need for rapid and reliable detection arises mainly from its volatility, as it is released during nuclear facility accidents as one of the first radionuclides. Okay. There is no sign. Breaking the wall involves the determination of iodine 129 from various types of matrices in similar manner, followed by its separation and determination. Implementation includes the selection of acidic or alkaline leaching of the samples, and comparison of different types of separation and detection techniques to achieve the highest possible yield of iodine 129. For separation, we used liquid extraction, extraction chromatography, and decomposition in an acidic environment. And detection techniques include liquid extraction detection, low energy gamma spectrometric system, and accelerator mass spectrometry. All analyses are developed or optimized at the Department of Nuclear Chemistry, with the goal of implementing them into the testing laboratory of radiochemical analysis, Faculty of Natural Sciences, Comenius University, Bratislava, which is accredited according to the international standard 17025, which is only one of its kind in Slovakia, and where I also work as the quality manager. My time is good. So we work with always with environmental types of matrices, such as sediments, pressurized grass, aquatic plants, or waters. For accelerator mass spectrometry, we collaborate with Nuclear Research Institute in Rzesz, and also with Czech Technical University in Prague. During experiments, our aim is to standardize the pre-treatment process of the samples. Thank you.
Thank you very much, Martina Bugriova. I apologize, from Comenius University, Bratislava. Any question?
Okay. Question. How original your approach is? Because detection of isotopes is a very broad theme. And there are many ways how to detect isotopes. So what is the original approach in your lab?
It is the step of the pre-treatment process, because we want to have it in a similar manner. So every type of matrices we want to do in a similar manner. So the pre-treatment process will be easier.
I see. So you standardized the whole process so you can have different...
Yeah.
Okay.
Thank you.
Thank you.
Thank you very much, Martina Bugriova. And now it is my pleasure to announce a presentation of Alžbeta Golianova from Comenius University, Bratislava, who is going to present her idea that breaks the wall of coffee's health benefits.
Thanks. Dear colleagues, it's a big pleasure to me show you a possible or how I can breaking the wall of coffee's health benefits. So. Coffee is not only an integral part of my daily routine, but also one of the most widely consumed beverages worldwide. Beyond caffeine, coffee contains a range of compounds, and some of which offer health benefits, such as chlorogenic acid, terpenes, micronutrient melanoids, and others. The resulting impact of coffee is influenced by a variety of factors, ranging from the plant cultivation to the method of coffee preparation. In our pilot experiments, we examined the effects of drinking coffee on red blood cells. Why red blood cells? The rationale for selecting erythrocytes lies in the fact that following coffee ingestion and absorption of its constituents in blood, erythrocytes are directly exposed to all biologically active compounds derived from coffee. Unlike other nucleated cells, erythrocytes lack repair mechanism, making them vulnerable to biochemical and biophysical changes when exposed to potentially harmful substances. And due to their short lifespan, they can be useful indicators of overall health. Erythrocyte poses the NK-atepase on their membrane, which in our experiments has demonstrated high susceptibility to modulation upon exposure to coffee. And we also found that drinking coffee affects the viscoelastic properties or deformability differently in various individuals. And based on these findings, I can confirm that coffee has positive effect on human health. But what needs to be adjusted is the type of coffee and the way it is served. And we propose a two-step process to determine if coffee consumption is potentially beneficial and or harmful.
And if the change in NK-AT path does not impair the passage of erythrocyte through capillar, coffee drinking is recommended. If it does, the individual who would like to continue drinking coffee should consider another method of coffee preparation. And I dare to say that coffee has the potential to have a positive effect on everyone, but it is important to tailor to consumption to individual needs. Thank you for your attention.
Thank you very much, Alžbeta Golianova. Any questions from coffee drinkers, maybe?
This sounds interesting. Are there any differences, inter-individual differences in elasticity of erythrocyte for those who do not drink coffee?
We had a big project for erythrocyte, and we found that NK-AT path is the main characteristic or what is influenced, but it was everything what is for us in this moment result or correct. What I can talk about or talking.
So you do not use controls of non-drinkers?
Yeah.
You use?
Yeah.
And do you see the differences in non-drinkers?
Yeah.
Yes, you do.
Thank you very much, Alžbeta Golianova. Our next presenter is Julia Horníková from University of Veterinary Medicine and Pharmacy in Košice, who will present her idea that breaks the wall of orthopedic gene therapy.
Thank you very much. So, did you know that orthopedic diseases are very frequent in our population, with more than hundreds of millions of people suffering with this disease in the whole world? Osteoarthritis is a degenerative and chronic joint disease, which is affecting young, but definitely in most part the old people. And there are still missing good procedures to help these patients. Also, there are many surgical treatments which are not definitely good enough to sustain and help regenerate the cartilage. So but there is an idea of biomaterial-guided viral vector gene therapy, which is offering potent alternatives for reestablishing the structural integrity of the matched cartilage. And this technique is already used in practice, but there are still many things that need to be checked and corrected. And because the viral vectors which are used need a lot of experimental tests, preclinical tests, which test their efficacy, durability, and safety. In this test are used a lot of experimental animals, which is not in alignment with 3R principles, which are protecting experimental animals, and they also take a long time. Here we come with a model of a chorioallantoine chicken membrane, which is an in vivo alternative model offering very fast results, is costly effective, and also it's very easy to manipulate. And we were already trying recombinant adeno-associated viral vector, testing them on this membrane. And this membrane is also in alignment with 3R principles, as it's not considered a living organism yet, the chicken embryo. And we were able to test transduction efficacy, then transgene expression, and also biocompatibility and safety in a combination with biomaterial, which is used in orthopedic regenerative medicine. These three factors belong to main factors which are needed to be tested in preclinical studies, as still the viral vector, there is a viral particle which can be dangerous for the recipient. And using the CAM model according to our results is very innovative and very supportive in reduction of animals which are used in experiments, as we can replace them with this model, and also we can get to much faster results in preclinical studies, and thus coming faster to results or to grow and follow with studies on people or clinical studies.
Thank you very much. I have to stop you. If you have any questions for Julia Horníková, please.
This might be good for testing the vector, but what about the gene that is in the vector? So which genes would you like to use for osteoarthritis?
Yeah, we are specializing in vectors which are carrying pro-argiogenic factors, as the problem with cartilage is that it doesn't have good blood supply. So we are putting for example, like VEGF factor or factors which can help them with angiogenic potential in combination with biomaterials also.
Thank you very much, Julia Horníková. And now it is my pleasure to announce a presentation of Natalia Chomova from University of Veterinary Medicine and Pharmacy in Košice, who is going to present her idea that breaks the wall of noxious commercial feed.
Thank you. Good afternoon, everyone. The sigma-sequence search in aquaculture and intensification of the production led to the worsening of water quality and required better and more effective therapy of the disease. To prevent losses and enhance production parameters, farmers start using antibiotics. However, overuse and misapplication of these substances is connected to the evolution and spread of antibacterial resistance and also accumulation of drug residues. Therefore, when the European Union forbade the use of antibiotics as feed additives, World Health Organization recommended the use of natural immunomodulators instead. Moreover, in countries like Norway, which is one of the biggest salmon producers, is fish protein in feed substituted with the soybeans, which is unnatural and may lead to the enterotitis, higher susceptibility to the infection, stress, and lowering immune response. To solve this problem, we have chosen probiotics. First of all, it was necessary to isolate and characterize correct autochthonous probiotic strain which will have good growth properties, will be able to survive in a fish intestine, and will be susceptible to the antibiotics based on EFSA regulation. Subsequently, we incorporated it into stable and economically affordable form for aquatic animals with various excipients. Probiotic feed prepared this way was later tested on healthy rainbow trouts, Atlantic salmons fed with soybeans, and also on fish infected with one of the most serious bacterial pathogens in aquaculture. Results of several experiments were similar. Reduction of pro-inflammatory response and stimulation of innate immunity was demonstrated. These results proved immunomodulatory potential of probiotics and its use in practice, which led to the submission of patent applications. One for application in Norway and the other for application in Slovakia and Czech Republic with the aim of commercializing probiotic feed in conventional farm breeding. Thank you.
Thank you very much. Thank you very much, Natalia Chomova. Any questions, dear jury members?
Yep. Yeah, that my word worked too.
No questions? Yes, Professor Calat?
Which probiotic strain is the best one?
Yeah, we tried several ones, but based on in vitro studies, we chose Lactobacillus plantarum and also Lacticase Bacillus fermentum.
Thank you very much.
Thank you.
Natalia Chomova. Our next presenter is Adam Kantor from Slovak Academy of Sciences, who will present his idea that breaks the wall of alien species monitoring?
Dear committee, dear guests, so I'll be speaking about biological alien species. So if you came for UFO, I'm sorry, but I hope it will be still interesting. So we use the term alien species to refer to organisms that spread beyond its original distribution due to human activities. And some of these species become invasive, and these cause huge problems associated with, for example, with biodiversity loss, human health, but also economic costs. As you can see in the top, these are the minimum economic costs of biological invasions calculated for approximately last 50 years, as reported in recent study in Nature. And managing alien species is also one of the Sustainable Development Goals presented by United Nations. So the most effective way of reducing these risks is by detecting the alien species in its earliest phases of invasion. At the same time, we have great citizen science initiatives in which people from public can partake in collection of biodiversity data. One of the most well-known is iNaturalist, in which there are currently more than 200 million of observations. So this is the map of number of observations in Europe. So we believe that among those large data sets, there must be very valuable information about spreading of citizen science, spreading of alien species, about spreading of alien species, but these data are not used very effectively now. So I came up with a procedure, which is quite simple, but it has not been used before, or at least not published before, which uses so-called checklists. A checklist is like a complete list of all species recorded in specific area. Almost all most of European countries have some checklist of at least one specific group of organisms. And as I am a botanist, I will be showing it on the example of plants in Slovakia, but this method would be applicable basically all around the world and on any other group of organisms. So what I did was that I took the observations of plants from Slovakia in iNaturalist, and I filtered out the observations of the species that are listed in national checklist of vascular plants in Slovakia. And I came to the much smaller sub-data set, which could be revised manually. And by visiting some of those localities in the field, I was able to find and confirm 4 new alien species for Slovakia. So these are the species that have never been recorded in wild in Slovakia before and could become potentially invasive. The most striking probably was finding of Lonicera japonica, which is the species that is considered, as highly invasive in America and also in many countries in Europe.
Thank you very much.
Thank you.
Thank you very much, Adam Kantor. Any questions of the jury?
Do you develop application for the citizens to record these findings, or how do you collect the data?
We are using this iNaturalist, which is a website and also an app, which is very effective, but it provides open source data for scientists and nature protectors. So we are using this data, but I just develop a framework for filtering it more effectively.
Thank you.
Thank you very much, Adam Kantor. Now it is my pleasure to announce a presentation of Jakub Leszczynski from Pavel Jozef Šafárik University in Košice, who is going to present his idea that breaks the wall of conversion storage.
Thank you very much. So today I would like to break the wall of conversion storage. If we talk about the conversion storage, in general, most of us imagine a bunch of lithium-ion batteries put together, and this imagination is not wrong because of the exceptional properties of lithium-ion batteries, which is high capacity, volumetric and gravimetric energy density of lithium-ion batteries in comparison to other types of batteries these days, for example, lead acid or alkaline batteries. They are suitable for storage of energy. But technology is still moving forward, and we start to hit the limits of lithium-ion batteries, and we need to develop another way how to store energy efficiently in big amounts. And that's why we are developing post-lithium-ion batteries, such as sodium-ion batteries, aluminum, magnesium-ion batteries. They are based on lithium-ion chemistry, but we change the material inside the battery. My research is mainly focused on lithium-sulfur battery. The properties of lithium-sulfur batteries are much higher than lithium-ion batteries. The theoretical capacity is a few times higher than of high-end lithium-ion battery. Also, because of the materials inside the battery, sulfur is abundant, is non-toxic, and pretty much cheap, so it's a big plus. Our research is mainly focused not only on developing the materials for the battery, but also on the optimization of parameters inside the battery. Our newest research, we try to develop the morphology of sulfur electrode. From the results of our experiment, we can already see that the battery that we created has a higher capacity than high-end lithium-ion battery. These days, the experiment that we are running, we are developing, or we are trying to study the ratio between sulfur, active material, and the electrolyte, so we can have a lighter battery. So thank you for your attention, and if you have questions.
Thank you very much, Jakub Leszczynski. Any questions?
So I'm just for 9 questions. I mean, it's impressive, 10 orders of magnitude, essentially.
Yeah.
But the question would be, how many cycles you can expect? Because this is one of the parameters that is also very important.
Yeah.
How many cycles you would expect to survive with this battery?
Can I run back to the... Okay. So from this data, we run 35 cycles of for each of battery, and in different speeds. So in the capacity for the best battery, that is here, the green one, we've got the capacity was higher at the end than at the beginning. So the active material starts working through the cycling of battery. So we can, we try 35, but if we can try maybe 100, the capacitance will go down just few percentage, maybe. Usually it's around 98% per in 35 cycles. So it's
very good.
Yeah.
Thank you.
Thank you very much, Jakub Leszczynski. Our next presenter is Kristina Licha from Comenius University Bratislava, who will present her idea that breaks the wall of DNA in small extracellular vesicles.
Okay, thank you very much. So let's look behind the wall of extracellular vesicular DNA, and maybe we will break it. But first, have you ever heard of extracellular DNA? It's a DNA outside of the cells that's floating in a blood circulation. It can be a good guy and works as a biomarker, or it can be a bad guy and acts as a damage-associated molecular pattern. These DAMs are released from stressed, damaged, or dying cells, which can trigger immune responses. And one form of extracellular DNA is its presence in extracellular vesicles, which are released from all cells. I know, I'm sorry, a lot of extracellular stuff. And this DNA is called vesicular DNA or evDNA. It can be stacked on the surface of vesicles or protected inside vesicles against degradation by nucleases. And these vesicles are very small, and they are involved in intercellular communication, or they can transport DNA from one cell to another, which can also trigger immune responses. However, evDNA is still a mystery, because it is not clear if and how much DNA is associated with vesicles. So in our research, we proved that vesicles from healthy human blood contain evDNA on the surface, stack on the surface, and also inside vesicles. And moreover, we found that 25 to 40% of DNA is protected inside vesicles. And the most interesting result was that mitochondrial DNA is more prevalent in these vesicles and protected against degradation. And why is it important that I mention mitochondrial DNA? Because mitochondrial DNA can also be a bad guy and can trigger immune responses. And these vesicles can act like a postman and deliver this mitochondrial DNA from one cell to another cell, which can lead to, which can potentiate inflammation and lead to development of autoimmune diseases. Okay, so thank you very much for your attention.
Thank you very much. Thank you, Kristina Licha. Any questions?
Extracellular DNA can serve as biomarker, so which kind of diseases could be diagnosed through this?
Most of the time it's probably cancer, which is diagnosed by this extracellular DNA, but it also we can use in, it's not a disease, but in a, I mean, you can diagnose when in prenatal diagnosis, this extracellular DNA also used.
Thank you very much, Kristina Licha. And now it is my pleasure to announce a presentation of Gabriela Oršová from Comenius University Bratislava, University Hospital Trnava, who is going to present her idea that breaks the wall of prostate cancer diagnosis.
Dear ladies and gentlemen, today I would like to present my topic, breaking the wall of prostate cancer diagnosis. According to the last data from OECD, the prostate cancer is the third common cause mortality of cancer among men. Risk of factors are obesity, older age, genetics, and race too. Management of patient with induced problem as blood in the urine, problem with urinating or erectile dysfunction starts with laboratory and radiology and diagnostic. And after positive results, patient undergo biopsy. But in my project, I would like to decrease the number of unnecessary prostate biopsies. Why? I would like to show you one non-invasive method which can define and grade prostate cancer. How we can do it. I want to show you radiomics. Radiomics is quantitative metrics in using non-invasive method, magnetic resonance imaging, with specific sequence, diffusion tensor imaging, DTI, based on Brownian movement of water molecules in tissue. We can, with this technique, specify and reflect microstructure tissue, and this sequence has also quantitative parameters too. One of these quantitative parameters is fractional anisotropy. Fractional anisotropy reflect biomechanical features, and thanks this value, we can apply these parameters to to detect predictor lesion in imaging, and then maybe use in future in clinical practice. Thank you.
Thank you very much, Gabriela Oršová. Any questions, dear jury members? Professor Calec?
So maybe a stupid question, but how much experience do you need for evaluation of this imaging, or is it somehow quantitatively analyzed by the instrument?
Yeah, it is only specific software where we make a range of interest, this lesion, and with help with doctor for this range, we define this lesion and quantify them.
Thank you very much, Gabriela Oršová. Now it is my pleasure to announce a presentation of Lucia Piatrikova from University of Žilina, who is going to present her idea that breaks the wall of histological stainings.
I am breaking the wall of histological stainings. Histological staining is a crucial factor in cancer diagnosis. AG staining is commonly used by pathologists, and it stains main tissue components. On the other side of the wall, there is immunohistochemistry staining, which provides more information, but is also more time and money consuming and not always available. Specifically, there is Ki67 staining, which evaluates the expression of Ki67 protein by Ki67 index. And now we don't know if or how is Ki67 information hidden in AG staining. So let's break the wall between these two stainings using machine learning. There is my proposed solution. The main part of this solution is a generative model, which can generate AG stained images conditioned on Ki67 index. Although generative models have already been applied in histology, this generative model is conditioned on Ki67 index. So it can generate AG stained images for a specific values of Ki67 index. And consequently, we can generate a sequence of AG stained images with a changing value of Ki67 index. And we can analyze how the change in the value of Ki67 affects AG stained tissue. And the second part of this solution is a deep learning model, which encodes a real AG stained image to our generative model, and it also estimates a Ki67 index directly from AG stained image. So we don't need Ki67 staining, so we can save time, resources, have reproducible results. And however, some other solutions exist where they also try to estimate Ki67 index directly from AG stained image, but we can, in addition to these solutions, using our generative model, modify original AG stained image to have different values of Ki67 index. So we can depict the progression of a cancer in this original AG stained image. And so this solution not just predicts the value, but also explains it in this way. And it can possibly, using different experiments, we can reveal the hidden relations between AG and Ki67 stainings, and thus break the wall between these two histological stainings. Thank you for your attention.
Thank you very much, Lucia Piatrikova. Any questions?
Is there any data or any intention to use it really prospectively or diagnostically in relationship to the progression of the disease and patient? Yes. Because this is just the one parameter, and progression of disease is not always predictive with this only one parameter. Are there any other attempts to use it?
We work with just this Ki67, and we collaborate with pathologists with this, and we analyze the results with them. So I'm not sure if I understand the question. We consider it only with this Ki67. But it can be applied also to other stainings, not just this one, if we have data.
Thank you very much. Lucia Piatrikova. And now it is my pleasure to announce a presentation of Alena Potočárova from Comenius University Bratislava, who is going to present her idea that breaks the wall of hidden health data.
Thank you for the introduction. So let's look at the water coming from our toilets. Wastewater is a great source of information about us, about our behavior. We were able to monitor coronavirus spread even 7 days before the outbreak. We can analyze the effect of antibiotics on bacteria present in a wastewater coming from the hospitals. And we can track narcotics to find out where is the place when they are made. And what about the DNA, human DNA? Is it present in a wastewater? In what form? Can we use it? Is it stable? Can we use it for a further analysis like a sequencing? Because human DNA is also a great source of information about us, about our health, about our behavior. So we looked at the water from our toilets in Bratislava, and we were successful. We found out that in the wastewater there is present a relatively high amount of DNA. Third of it is a human source, and almost half of it is protected from the degradation. So we can say that it's stable and representative enough for the analysis like sequencing. And what can we do with this great source of great pack of available data? We can look at the incidence of diseases across the population, like what is the incidence of genetic mutation specific for autistic disorder in Bratislava, or we can find the connection between the environmental pollution from a local factory and genetic mutations in people living nearby. Or we can also look closely to the human migration pattern to focus more locally and to look more closely on the behavior of people living thousands of years ago. So, thank you very much.
Thank you very much. Have you got any questions? Professor Buzek?
When we worked on this project, also I've looked at the PubMed in these days, and there are all of the articles are focused only on the DNA from a bacteria or different pathogens, how they are changing, how they are pathogen, and none of the articles were focused on a normal human DNA. So we didn't find anything similar. And how we can use it, as I have mentioned, we were also trying to go a bit further to use it in a sequencing, but there are some small other steps to rule out the effect of inhibitors of sequencing and so on, but we are already working, or we're working with our colleagues on also these steps to put it a bit further.
Thank you very much.
Thank
Alena
you.
Potočarová. Now it is my pleasure to announce a presentation of Miroslava Šimiaková from Comenius University Bratislava, who is going to present her idea that breaks the wall of cancer gut microbiota.
Dear audience, today I would like to present to you the idea how to breaking the wall of cancer gut microbiota. The treatment, such as antibiotics, chemotherapy or radiotherapy on acute lymphoblastic leukemia in pediatric patients, disrupts gut microbiota and persists in remission. while pathogenic bacteria such as Clostridium difficile on Enterobacter on the gut of pediatric patients demonstrate antibiotic resistance, probiotic promotes beneficial shift on the microflora. We also know about probiotic effects of dairy products such as yogurt or about probiotic capsules. However, Slovakian brinza cheese is acknowledged for its probiotic benefits when consumed as a food. However, it also contains coliform bacteria, which poses potential risk for a patient with acute lymphoblastic leukemia. The solution is isolating and culturing probiotic strains from brinza cheese, for example, Lactococcus species or Lactobacillus species, followed by lyophilization and encapsulation, to administration firstly for health control groups, and if we will have positive results, then administrating to pediatric oncology patient in remission. This approach minimizes of the risk adverse effects such as bacteremia, while facilitating restoration of gut microbiota in pediatric oncology patients. The restoration of gut microbiota positively impact on physical, mental, and metabolomic health of pediatric oncology patients. Thank you very much.
Thank you very much. Any questions, Professor Pastoreková?
This approach has been used already for the adult patients. What is the difference or innovation concerning pediatric patients?
So, pediatric patients have problem with gut microbiota, and also we focus this topic to leukemia, and we know leukemia is often in a pediatric age. So I think this is the answer.
So it's the conditions, but in principle this is not a new approach, so to say. At least what I know, I don't know. Thank you.
Okay, thank you.
We still have 15 seconds, so if there is any other question, we still have some time to answer it.
So you have chosen brinza because we are in Slovakia, but what about all the other cheese available in France or Switzerland or anywhere else? Are they bad?
So you said we choose Slovakian brinza cheese because it's our national food, and I think it's really interesting to work with this food.
Thank you very much, Miroslava Šimiaková.
Thank you.
And now it is my pleasure to announce a presentation of Imelda Oktatampu-Bolon from Slovak Academy of Sciences, who is going to present her idea that breaks the wall of her electrocatalysts.
Thank you. So, global warming is here, and it's a nightmare. We use fossil fuels 15 billion metric tons per year, and it costs us a lot. So, it increasing the level of the CO2, damaging our environment and risking our health. So, to combat this, we have to come with the new solution, clean energy, greener energy sources. And we can achieve this through the water splitting that produce green hydrogen, which is 100% sustainable and pollution-free. However, it costs a lot, $4 to $6 per kilogram. It's even 3 times higher than the fossil fuels. Why? The issue is from the materials, the electrocatalyst, and the process. Usually, we use a noble metal, the rare one. So here we come with the new solution, providing the nickel sulfide because it's abundant. Yeah. However, there is an important issue at the same time. So the synthesis of the nickel sulfide is always involved with the multi-step reactions, high temperatures, toxic chemicals. So for example here, the nickel nitrates and oleoamine already known for its carcinogen. So here we come to break the walls of HER electrocatalyst by simply applying the mechanochemistry. So with the very simple one-step reaction without solvent, so we successfully synthesized the nickel sulfide in our lab by the specific procedure so-called mechanically induced self-propagating reaction. So this is a combustive-like reaction. That's the evidence. And within seconds, we successfully synthesized this nickel sulfide in the form NES and Ne3S2. And we evaluate the electrocatalytic, and it's quite promising. So, yeah, we hope with this we can contribute to the goals of the SDG number 7, number 11, and 13. And we hope that we can contribute more for a better climate, better lives, and provide sustainable energy and reach the goal of 2050 net zero greenhouse gases. And thank you.
Thank you very much. Thank you, Imelda.
Thank you.
Any questions, please?
So this is a mechanochemistry.
Mechanochemistry.
You need a lot of energy, I guess, to really do this milling and other processes.
No.
So what is the balance of? Because you produce pollution when you generate energy for your process.
So actually, basically, we just use very less energy because the concept of the mechanochemistry is mechanical forces. But just applying the mechanical forces, let's say the reactions could be proceed. So very less energy that we can use. Yeah. Yes. Thank you.
Thank you very much, Imelda Oktatampubolom. And now it is my pleasure to announce a presentation of Patricia Vaňová from Technical University of Košice, who is going to present her idea that breaks the wall of bridge maintenance.
Hello, everyone. So, as it's been said, my name is Patricia Vaňová, and I would like to break the wall of bridge maintenance. Now, we all hate driving on roads under construction, right? And it feels like it's happening more and more often. In the Slovak Republic, over 34% of bridges are in poor to critical condition. It's very clear we have a growing problem. The current approach to bridge maintenance, visual inspection, is both costly and insufficient. A newer method called structural health monitoring uses sensors and data to assess the bridge condition. However, it relies on having the data from the bridge when it was in the undamaged state, right after it was built. This doesn't help with the growing number of aging bridges, which are deteriorating and posing greater risks to both our safety as well as to our patients. What we need is a comprehensive solution that can continuously monitor the health of a bridge, even without requiring the data from the bridge when it was in the original state. And that's where my research comes in. We aim to create a system capable of assessing bridges in real time under diverse weather and traffic conditions. To achieve this goal, we are building a vast multi-damage database of over 5,000 damage cases through numerical modeling and analyzing this data using statistical methods, modern statistical methods, and artificial intelligence. The created system will enable us to identify potential issues early on and take proactive measures, ultimately preventing sudden bridge failures before they occur, avoiding costly repairs, minimizing disruptions, and ensuring continuous safety and reliability of our infrastructure. And this research has the potential to benefit the infrastructure not only in Slovakia, but all around the world, improving safety and reducing costly maintenance delays for everyone. While this may not stop all the construction signs from popping up from time to time, at least we can rest assured the bridge will remain safely in place while we cross it. Thank you. Do you have any questions?
Thank you very much, Patricia Vaňová.
I'm asking questions all the time, doing the job of the jury. Anyway, so yes, it's very interesting, and certainly it's not applicable only to bridges, but definitely for any structure. But important component of this are the sensors.
Yeah.
And I am just wondering, what do you monitor with these sensors? Like, Because essentially, I think that you have to look for micro cracks, fatigue of the material, the vibrations that are kind of untypical vibrations. So, what type of sensors and how would you collect data from your bridges across the country to some central body that monitors?
We are using accelerometers. So accelerometers are used to identify the natural frequencies and natural shapes of the bridge. So we are using the dynamic behavior of the bridge to see the differences in data. And there are many bridges in the world that have not only accelerometers, but a lot of sensors put on them. Just the data is not used, and we cannot for sure right now say like there is something happening. But there are systems even, not so much in Slovakia, but even in the Czech Republic that monitor bridges continuously. They just don't know
how to use the data. So
yeah.
It's essentially your innovation is the using the AI, whatever that means in this context, to really to analyze data more efficiently and to follow.
Yeah, to have a more complex research and to have more data to analyze.
Thank you. Thank you very much, Patricia Vaňová. And our 17th presenter is Nitya Hannah Wilson from Tomáš Baťa University in Zlín, who will present her idea that breaks the wall of EMI shielding materials. Welcome.
Hi, everyone. Before moving to my topic on breaking the wall of EMI shielding material, let me ask you a question. Have you ever experienced network issues while making phone calls? I believe most of us. Have you ever thought why this is happening? Let me introduce you to one of the emerging concerns, which is termed as electromagnetic interference, or in short, EMI. It sounds like a phrase from a science fiction movie. Perhaps it is not that. It is something which is actually affecting our everyday lives. As the tremendous increase in the usage of electronic and communication devices, we all know that it is affecting us by the harmful electromagnetic radiations. Nevertheless, most of us are just unaware of this concern, which is called interference, which is called EMI. So, what impact does this EMI have? Due to the signal losses, it can probably lead to the malfunctioning of electronic devices. Besides, these radiations are harmful for our health as well. Now you must be wondering, aren't there any solution for this problem? Of course, there are some traditional metal and metal-based EMI shields, but somehow they have some limitations, which can be seen here. So, what is the solution? I hereby introduce a simple, desirable, very reliable, and innovative approach by using a conducting and magnetic fillers in a suitable polymer matrix, and through melt mixing process, after that stacking layer by layer to form a multi-layered polymer nanocomposites, which can be seen here. And I have here is a developed material I have here with me. And we have used graphite and cobalt ferrite nanoparticles as conducting and magnetic fillers on thermoplastic polyurethane matrix. This has shown effective EMI shielding efficiency of around 54 dB, thus making it suitable for commercial applications. I have also studied the possible mechanism of EMI shielding, and second part of my research will be focusing on making this material transparent and also fine-tuning into specific frequency. Thus, hereby, I declare that we have developed a material which can be used, which can be applied by this sandwich structured model, could be used as an effective EMI shielding material. Thus, I could break the wall of conventional EMI shielding materials. Thank you.
Thank you, Nitya Hannah Wilson. Any questions, please? Professor Buzek.
Yeah.
They are relaxing here, the rest of the jury. So I'm really curious because electromagnetic interference,
Yes.
I mean, we know how to protect, how to shield with the Faraday effect. We can
Yes,
shield.
yes, Faraday cage.
Exactly, exactly. Now, how can you shield? So it means you can be rather sort of whatever security safe from the interference if you are inside the cage. But then essentially, if I want to communicate outside the cage and not be interfered by other electromagnetic sources, how do I do that?
That is going to be my second part of research. Actually, this sample, we can incorporate the same materials in your fabrics and many things, many methods of implementation of these things. And we are planning, my second part of research will be specifically in tuning of this, self-tuning of this to specific frequency and also making into a transparent material. So for that, phone applications, mobile, and these things.
Okay.
So it is going to be a second part of research. I'm just in the second year of PhD now, so it will be continued with.
I can imagine many applications.
Yes, yes, definitely. It is going to be helpful for all of us. So I thought it will be a great platform for me to introduce because most of us are not aware.
How original your research is? Sorry.
It's actually going to be patented. Means the idea, especially from this graphite and cobalt ferrite nanoparticles, which can be simply prepared with sonochemical method in any labs, any chemistry labs, and can be practically mixed easily. We have other techniques as well, like vacuum filtration, acid step techniques, but this is going to be the simple, reliable ones. And already we are in the stage of processing it. So thank you.
Thank you very much, Nitya Hannah Wilson. Dear Falling Walls Lab participants, thank you for your interesting presentation. And now it's time for the members of the jury to evaluate your presentations and tell us who is the winner, who will represent Slovak Republic in the final in Berlin. During this time for the jury, which will take approximately 30 minutes, something like this, I invite you, dear guests, dear participants, for a refreshment break. So see you here around 30 minutes. Dear participants, dear competitors, dear distinguished guests, dear members of the jury, so it's time to announce the winner of Falling Walls Lab Slovakia 2024. Therefore, I am happy to have on the stage back the jury chairman, Mr. Buzek, also Professor Mozzo and Susanna Weziek from DAAD. So let's start. We are excited to hear who is the winner.
Thank you very much to all of you who participated in this afternoon competition. It's so refreshing to know that there are young researchers who are really interested in research, in doing research in Slovakia. And I'd like to remind you, this ticket to Berlin is a return ticket. There's a problem in our country. So whoever will win, whoever will win this competition, I know who will win, please remember, you have to come back and to help this country. So that's first. We had a lot of discussion, as you noticed that it took us longer than 30 minutes. It was not easy. I mean, we have to take into account many criteria and also like how a person would attract attention of the general public in Berlin. So I'm not allowed to tell you the results. Those will be told by Peter Mozzo. But I just want to convince to you that even if you are not among the first three winners, you all have won. I mean, you all have contributed, you all participated, you have new experiences, you know how difficult it is to give a 3-minute elevator pitch speech, how to attract other people to your research, and probably now it's time to pass microphone or whatever.
So it was really not easy. And as you very well know, we are not polyhistors, so even we could make a mistake, but we try to avoid making mistake. Thank you all participants for valuable presentations, for their enthusiasms, for their contribution to this event, and for coming. I will now announce three finalists. Please come as I announce you. So our bronze finalist is Jakub Leščinský from Pavol Jozef Šafárik University in Košice. For his contribution, breaking the wall of conversion storage. We will give you gifts all together. Please, yeah. Our silver medal finalist is Katarina Bilská from Comenius University in Bratislava. For her contribution, breaking the wall of biofilm-associated infections. And our gold finalist is Adam Kantor from Slovak Academy of Sciences. For breaking the wall of aligned species monitoring. Congratulations, and I will just briefly comment what you will get. So the gold medalist will get return ticket to Berlin, to the Berlin final. And maybe you remember that we explicitly asked you for each presentation to say clearly what is the difference or what is new in your presentation or what is in your result compared to the rest of the world, to the state of the art of knowledge in your problematics. So this is something which you have to better formulate next time. So each of you will get our book, Earthquakes, Tragic Challenges in History. Why? 1 year ago, we gave a book which was co-authored by bronze finalist of Falling Walls Slovakia, Klaudia Kyselicová. This year, we are giving you to three medalists a book which is again co-authored by the bronze finalist of Falling Walls Lab Slovakia. So this is a kind of informal challenge for the next years. Please write books and we will deliver them to Falling Walls Lab Slovakia finalists. And of course, each of you will get some small gift from the institution represented by Susanna. So congratulations. Time for joint photograph. But maybe before that I just make one comment. At the beginning I commented. It is the first time I see the ratio 14 to 3. The lady is really dominating the participants. And now we felt a little bit uncomfortable. So I want to comment that we absolutely were impartial. We were absolutely impartial regarding the authorship. That is now. I just want to be politically correct and that stuff. So I want to just to convey the message that the selection was really unbiased, completely unbiased, according to the presenter. So we honestly think, as based on evidence and the facts, that this is. That in spite of this ratio, because 2 of the 3 men are on the stage, so it is like very unusual for us statistically, but that statistic doesn't count here because it's honest decision of the jury.
that is one comment I wanted to make, that everybody's comfortable with this selection. Thank you.
This concludes our ceremony. Everybody is welcome to take more refreshment. Thank you very much for coming and for supporting young scientists and innovators and for supporting our future in Slovakia.
Thank you very much. It's clear that this event is coming to its end. Thank you very much. And let me congratulate all the participants and all the winners. And let's hope you're going to represent Slovak Republic very well in Berlin. We are crossing fingers for you. Ladies and gentlemen, dear distinguished guests, please let me invite you cordially already for Falling Walls Lab Slovakia 2025 next year. And before the route starts, I would love to ask the participants and the members of the jury to gather in front of the photo wall outside in the lobby to make a joint picture as a souvenir of today's event. Thank you very much and see you next year. Goodbye.